Hello, everyone. Welcome and thank you for attending our first sessions of the EMN live webinar series. My name is Kim Boon. I am currently a PhD student in University Malaya, Malaysia. And on behalf of the Early Member Network Committee of the Metabolomics Society, I will be co-hosting this event with Michelle Reed, a scientist at Crystal Therapeutics in Maastricht in Netherlands. Thank you again for joining us. If you would like to hear this talk at a later time, the webinar will be uploaded to the Metabolomics Society website with a link distributed through our social media networks. We encourage you to connect with us online via Facebook and Twitter for future content, and feel free to email us with suggestions or input. Today, we have two talks. The first from Professor Peter Dorrestein at the University of California, San Diego. His talk is entitled Annotating Unknowns in Untargeted Metabolomics Data. Professor Peter is the director of the Collaborative Mass Spectrometry Innovation Center and a co-director, Institute for Metabolomics Medicines in the Skate School of Pharmacy and Pharmaceutical Sciences and Department of Pharmacology and Pediatrics. Although born in Utrecht, the Netherlands, Dorrestein did his undergraduate in metallo-organic chemistry at Northern Arizona University, under the guidance of Prof. McDonald, and his graduate work in mechanistic enzymology at Cornell University under the guidance of Prof. Bagley. He performed his postdoctoral work as an NIH NRSF fellowship in top-down proteomics in the lab of Prof. Kelleher. Since his arrival at UCSD in 2006, Prof. Dorrestein has been pioneering the development of mass spectrometry analysis ecosystems for the community and methods to study the chemical ecological crosstalk between microorganisms, including host interactions for agriculture, diagnostic, and therapeutic applications. He is on the scientific advisory board of CIBEL and is a co-founder of Ometa and Anveda. In his spare time, Dorrestein likes to spend time with Kathleen, his better half, and his awesome daughter Tatiana. He also likes to garden, spend time in the mountains, rock climb, hike, kayak, and mountain bike. Our second talk is from Emily Gentry at the University of California, San Diego. She will continue the talk, Annotating Unknowns in Untargeted Metabolomics Data. Dr. Emily Gentry received her Bachelor of Science in Chemistry from the University of North Carolina at Chapel Hill in 2013. She then obtained her PhD in Chemistry from Princeton University in 2018, where she worked in the lab of Prof. Rob Knowles, developing new methods for the synthesis of pyrroloindolene natural products. She is currently working as a postdoctoral researcher in the lab of Prof. Peter Dorrestein at the University of California, San Diego, where she is developing a new strategy using synthesis and mass spectrometry for the annotations of unknowns in untargeted metabolomics data. Outside of the lab, Emily enjoys yoga, golf, and walking or hiking with her dog, Moira. Before our talks, I would like to announce that the next 2022 webinar will be on 17 February 2022. Professor Kati Hanhi-Neval from University of Turku and Dr. Ville Konstinen from University of Turku and University of Eastern Finland will discuss multifaceted relationship of diet and health examined with proteomics approach. Audience members, remember to submit questions throughout the presentation in the chat and include who the question is directed towards by indicating the speaker's first name followed by the question. Now, I will give the presenter control to Professor Peter, and the platform is now yours.
Thank you very much. Let me close this one. People can see you. Great. Well, thank you for the EMN for inviting both me and Emily to be a speaker here today. To me, it's really exciting about talking about a topic that is actually really difficult to carry out, particularly for early members, and that is the annotating unknowns. And so I'm going to carry you back to 2016 a little bit. And so if you would have taken a human biospecimen or a plant specimen or an ocean-derived specimen, and you were to subject these to LC-MS/MS-based metabolomics, you get all these fragmented spectra. You would then search against these reference libraries that were available at the time. And the average annotation rate at that time was 2% of the information. That meant that in general, we weren't using 98% of the information. Now, fortunately, the community has gotten better in sharing their data, sharing their annotations, sharing their libraries. And now this has grown to 10% of the data that gets annotated. And so it's a really significant improvement. It's a 5-fold improvement, but it still means that 90% of the data remains unannotated. And so today, what we're going to highlight are 5 different methods that we use within the context of the laboratory, or developing in the context of the laboratory, to be able to begin annotating some of this unknown space that you can find in untargeted metabolomics. And the tools that we're going to describe are molecular networking, and then using molecular networking to create a new library we call the nearest neighbor suspect library. And then some annotations are, so up until now we talked about structural annotations, but there may be other types of annotations. And so 3 and 4 really describe how we're going to use repository information to provide insight into this unknown metabolomic space, and then Emily will describe reverse metabolomics. And so molecular networking is a foundation of a lot of the things that we're going to talk about. So imagine your sample. You have a large diversity of molecules that are present in your sample. You subject these to untargeted metabolomics analysis, and now you have a large number of MS/MS spectra that reflect the molecular composition of your sample. What we do in molecular networking is we generally merge all identical spectra by using some sort of clustering algorithm, and there's a number of different clustering algorithms that exist. One of those is MS cluster. Another one we recently developed is Falcon. But once you apply MS cluster, you now have merged all the identical spectra, and you have a picture of all the unique spectra that are present in the sample. And we often represent these as nodes, shown right here. And then we do a spectral alignment. So we don't look for identical spectra, but we look for relatedness within the spectra. And that allows you to build this network. And the way to think about molecular networking is that this is really a data visualization strategy that then reflects the molecular composition of your sample. And this visualization really aids in the interpretation of the sample contents. And so let me share, because many of you are not yet used to looking at molecular networks, and some of you probably are, but many of you likely aren't. So I'm going to carry you through an example of how we've used molecular networking to propagate annotations. And so here's an example by Robbie Quinn, a former postdoc in the laboratory. He's now runs his own laboratory. But he had the question about how much does the microbiome really impact the chemistry of the host. And so the way he studied this was he took germ-free mice, so mice that don't have any microbes, and then mice that were colonized with a normal microbial community. He subjected these to extraction and then LC-MS/MS-based analysis. And he created molecular networks with them. And so what is nice is it creates this visual picture of some of the molecules that are present. And then you can color code it based on the data type. So SPF are specific pathogen-free mice. Those are mice with a normal microbial community. And then in pink, we have the germ-free mice. And so what he was looking for were nodes that were only found in the mice that were colonized with a normal microbial community. And all he has to look for are nodes that are blue. And so in this little cluster right here that is shown on the left side, you have 3 nodes that are in blue, and then you have other nodes that are only found in germ-free mice, and then you have 1 node that is found in both of them. And so fortunately, he had one annotation, and that was a spectral match to the reference library to glycolic acid. And so what is nice about glycolic acid in the positive mode, if you fragment it, it cleaves right at this amide linkage, and you get almost like a B and Y ion type scenario that you find in peptides, in the fact that we see a glycine loss, and then you see glycine itself in the MSMS spectra, and the other ions are characteristic of this cholic acid moiety. Now, what you'll see is adjacent to this, we see a 90 Dalton shift, this node that is blue. And if you've ever done any mutation analysis in proteomics, you might recognize that 90 Dalton is a glycine to phenylalanine substitution. And so we hypothesized that this phenylalanine was attached to this cholic acid, so we synthesized it, and we were able to verify this with the synthetic standard. And then adjacent to this, we had a 15.996. If you look at the periodic table, that matches up to oxygen. And so this is an addition of an oxygen. And it turns out to be that it's tyrosine conjugated bile acids. So these were new bile acids that we discovered by simply beginning to answer these questions, but really using molecular networking to propagate those annotations. So this is how you can go from a known molecule to gain insight into many of these unknowns. But then Wout and Ming came along, and they had this really crazy idea, because what I just showed you was that we could propagate from a known molecule in germ-free mice to an unknown molecule in mice that were colonized with a microbial community. What if we do this at the repository scale? In other words, all the untargeted metabolomics data that's publicly deposited, if we take all their MSMS vector, can we then begin to create a library of propagated molecules? And so right now there's about 1.4 billion MSMS spectra that are in the public domain, if you take all the public data sets. And so they wanted to create a molecular network that had 1 billion spectra in it, or more than 1 billion. And so what that meant was that you needed to make a comparison of 1 billion spectra to 1 billion spectra. Well, that becomes computationally very challenging. So they needed to come up with a solution to be able to actually make this comparison and build this network. And so what they came up with is that they basically built molecular networks for each of the data sets independently, and then combine them. And so this allows you then to take one annotation from one data set and propagate the annotations to another data set if that reference spectra is not available. And so here, for example, we have a 14 Dalton shift. And if you then look at the MSMS spectra, it's attached to this acetyl group. And so that becomes a propionyl carnitine. In other cases, all we can assign is a molecular formula. But what we now do is we create an entirely new library with all these new suspects and try to provide explanations for it. And the question, does it really work? So here you have hexanyl carnitine. This is a C6 carbon with 0 double bonds. So if you count the number of carbons on this fatty acid chain, those are 6 carbons, and there are no double bonds in this fatty acid chain attached to it. And so then we had a suspect library that matched to hexanyl carnitine, but that was 2 Dalton smaller, and in fact it's 2.016. To anybody who studied metabolism, this suggests that there's a double bond introduced where there was previously a single bond. And so now what we have is we have this same azocarnitine. However, now we have 1 double bond. We may not know the location of the double bond, but the suspect library does give you a lot of insights. Here we have hexanyl carnitine minus 12.036 Dalton. Now, some of you may look at the periodic chart and say, oh, that's carbon. No, carbon is 12.000 Daltons, and so it is not a carbon loss. So what's going on here? Well, if you try to come up with what makes up a 12.036 Dalton modification, what you will see is that it has to be a gain of oxygen and then a loss of 2 carbons and 4 protons. And that essentially becomes a hydroxybutyryl carnitine. However, we even know the position of this hydroxyl group, because if the hydroxyl group is in this position, and this is only something that an expert can recognize, you have this ion at 145, and that's what we see here. So now we know at least that it's a 3-hydroxybutyryl carnitine. We do not know the stereochemistry of this hydroxyl group, so it still leaves us with 2 possibilities, but it still gives you a lot of information. So right now the suspect library has literally hundreds of different azocarnitines that have been observed in this fashion. However, the question that we had is, can we also look at more complex molecules? And so this is one example that Nicole Avalon has been studying. She's been looking at aprotoxin. This is a small molecule that is isolated from cyanobacteria, but it's a fairly complex molecule. And the reason why she's interested in it is because this has been in multiple phases of clinical trials as an anti-cancer agent. And so she wanted to find analogs of these aprotoxins so she can, in the future, do structure-activity relationship type studies. And so you can now begin to use this suspect library to find these analogs and then make a hypothesis or build a hypothesis of what the structure might look like. And so here, for example, here the difference between the green and the purple is really a methyl group. And if you start to look at it biosynthetically and really pay attention to all the fragmentines, you'll find out that that methyl group is lost in this modified tyrosine analog. And so now the most likely location of this loss of that methyl group is methoxy tyrosine. And so the suspect library itself, if you use it together with all the compound libraries that are available, it actually increased the number of annotations 2-fold. So we get a pretty significant increase in the number of annotations that we get with this suspect library. That's pretty exciting. But what do we do with the remaining 80%? There's still a lot of data that we cannot explain. And we really struggle with this in my laboratory, but anybody else throughout the world. And so Ming had this idea, well, what if we, instead of provide structural annotations, what if we can leverage repository scale information to begin to annotate unknown information in these untargeted metabolomics data sets? And so this is the first example of a tool called MASS that Ming developed to be able to do this. What does MASS do? Well, if you take this global molecular network and you have an unknown MSMS spectrum, you can search this global molecular network and then not report, unless the structure is known, not only report the structure information, but you might be able to give insight in terms of the phylogeny, so taxonomic identification. You may be able to find age-related questions. So that MSMS spectrum may have only been found in older samples and not younger samples, for example. It may be associated with disease. It may be different tissue types, different biofluids. It may have to do with sex distribution. If you're looking at environmental samples, you may get longitude, latitude information, or altitude, depth information, mass spectrometry ontology information. So even when you cannot get structural insight, you can get potentially a lot of insight about this unknown molecule. And this turns out to be very, very powerful. So for example, in this case, we had these spectra that we had identified as phenylalanine conjugated cholic acid and tyrosine conjugated cholic acid, but we were finding these in mouse models. And so often when you do mouse model studies, people want to know, is it translational? Is it translational to humans? And this is now a pretty easy question to address using MASS as a resource. So we search this MSMS spectra against the repositories, and all of a sudden, we start to see these same spectra in a lot of human samples. Not only do we find them in human samples, we see them upregulated in samples associated with IBD, cystic fibrosis, diabetic infants. And so with one search, we actually learned a lot without having to actually go in and collect human data ourselves. So it's a pretty powerful piece of information. We're starting to build topic and domain-specific masks, and I'm happy to collaborate with anybody on this. But Kiana West and Robin Smith and Ming teamed up to do this. But now when you search in MSMS spectra, and if it's found in food, you may be able to get the food distribution. So in this case, you may have an MSMS that matches to fruit and that's, let's say, animal sources. And it may end up only be found in berries, and then you may find some specifics about the berries. So even if the structure is unknown, you still get a lot of insight in terms of the distribution of where these type of molecules are found. So this is, to me, really important and really sort of a future way of thinking about annotating untargeted metabolomics. But the ability to organize a single molecule and how it's distributed among the different foods turns out to be quite powerful. And that leads us to reference data set driven metabolomics-based annotations. And that is basically, if you take reference data of foods, so in this case, we have about 3,600 different foods that are available in the public domain. Hopefully over the next 5 years, 10 years, that will be 100,000 or 1 million different foods that are sampled. And then you can take clinical samples and you can co-network all of these. And then instead of reporting structural information, so annotations of molecules, instead what you report is the frequency with which this food ontology has been observed. So you get this data table where you have samples on the x-axis and then food ontology on the y-axis and then frequency or intensity with which it's observed. And that data table is so important because that allows you to then do downstream analysis. And so that's exactly what Kiana West did with this IBD cohort. And so here what we see are in circles, patients that had Crohn's disease, in triangles, healthy controls, and in squares, patients with ulcerative colitis. And then she color-coded here with the readout for fleshy fruit. And what you can see are the people on the left have more spectral matches to fleshy fruit. The people on the left have more spectral matches to vegetables. And whereas the people on the right have more spectral matches to meat. And on the top, you have people who have more spectral matches to fermented grapes or wine. And so once you have this organized, this PCA pattern organized based on diet variation, now you can do regression analysis and see how do these patterns relate to clinical phenotypes. And so when she did this, well, one of the things that she observed was that there was no dietary pattern observed in relationship to disease, meaning patients with ulcerative colitis, Crohn's disease, or the healthy controls, they don't have a different dietary pattern. Now, patients that have different ages, they also don't have a different dietary patterns. Male versus female do not change. There's no observable dietary pattern change based on this readout. However, if we now look at the microbial diversity based on the sequencing that was available, so microbiome sequencing, now we can start to see that there's a much higher diversity along the PC1 axis. In other words, where there's more spectral matches against fruits and vegetables, you see a higher diversity of these microbial communities. And this has been reported in the literature before, where people who have consumed larger amount of fruits and vegetables have indeed a higher diversity. And now we look at disease activity or disease severity. So how terrible are these patients feeling, or really actually, there are actually clinical diagnostics associated with this. And the disease symptoms worsens if you go to the right on this PCA axis. In other words, the more people consumed red-type meats, the worse the symptoms were in both Crohn's disease and ulcerative colitis. Now, what was really interesting is you now look at the PC2 axis, the more fermented grapes spectral matches we had, the less severe the disease. So there may be some protective function that might be present in grapes or wines. And so I want to leave you with a few more take-home messages before Emily starts her talk. And that is, we really need new ways to annotate the unknowns. And I think the early career members are going to actually have a big impact on this over the next 10 years. The next 10 years will be revolutionary of how much data we can annotate in untargeted metabolomics. Really, one of the things that we should become better at is leveraging repository information. There's a really remarkable hidden resource that is not widely used, and more people need to start using these resources. So Metabolomics Workbench, Metabolites, GMPS Massive, these are three major resources that people should really begin to leverage and download the data that exists and begin to build better annotation tools. And really, I want to encourage you to share your data. And the reason why I say this, right now, I would imagine about 1% or less even of all metabolomics data gets shared in the public domain. And yet, that 1% is responsible for this growth curve from 2% in 2016 to the 10% of direct spectral annotations we have now. And we've seen with the suspect library, we've now been able to grow that to 20%. But if everybody were to share their reference spectra, annotations, and data publicly, we're going to have a really big impact really quickly. And of course, this work is not done in isolation. We recently looked at it, and I have 1,400 co-authors in the last 5 years alone. It's a really large community that has been able to contribute to the resources that we've built. But I want to pull out one particular individual, and that's Ming Wang. So for anybody who uses these public interfaces, Ming has been the key person that has been developing tools such as GMPS. And so I want to really thank all the collaborators, but also funding support for enabling the resources that we've been building. And then finally, before we take any questions, we'll take questions at the end of Emily's talk. I will pass the baton on to her.
Thank
Emily,
you,
it's your
Professor
turn.
Peter. Yeah, thank you for a great talk. So audience and members, there are questions in the chat. Please keep them coming in. I am now transitioned to Emily Gentry to further dive deeper into annotating unknowns. Emily, the floor is yours.
Perfect. Thank you for unmuting me. Sorry about that. Yeah, so thank you everyone for being here today. I'm going to keep my video off for the presentation, just so that I have enough bandwidth. But thank you also to the organizers for putting this all together, and thank you for Peter for introducing what I'm going to talk to you about today, which is that I'm excited to share with you a new reverse metabolomics strategy that I recently developed during my time in the Dorrestein lab. And it combines synthesis and mass spectrometry to discover new small molecules in humans and other animals. And this project was really the result of my love for chemical structures. So to give you some background, as you heard in my intro, I did my PhD in synthetic organic chemistry. And that's where I really developed a love for chemical structures. And then I went on to join the Dorrestein lab for my postdoc, where I'm currently at. And I wanted to learn more about untargeted mass spectrometry, because I was really interested in better understanding how small molecules are involved in human health and disease. But then once I joined the Dorrestein lab and started actually working with untargeted mass spectrometry data, I was really shocked to see just how few ions were identified with chemical structures in this type of data. So to me, the results from an untargeted metabolomics experiment looks like a blurry picture where you might be able to kind of get a sense of what's going on. You know, you can maybe see the outline of a figure here, but it's really challenging to draw conclusions from these types of images. And so to give you a little bit better context and to reiterate what Peter presented, so in a typical untargeted metabolomics experiment, we'll see anywhere from 2 to 3% annotation to 15% annotation, meaning that this is the number of ions that had spectral matches with library spectra. And so here, of course, the annotation rate depends on the type of samples that you're analyzing, as well as the number of samples. But overall, the overall message is the same, which is that the majority of ions detected in untargeted metabolomics experiments remain unidentified. And so this gap in knowledge is what drove me to build a new workflow that aims to deblur the picture in untargeted metabolomics. And today I'm going to share with you a story about how I've applied this method to begin to deblur the picture associated with bile acids. And so I was really interested in first targeting bile acids because these are really seemingly simple steroidal compounds, but they have very complex and important roles in humans and also other animals, like my dog Moira, which was actually, that figure in the blurred image. And so although bile acids were originally discovered for their role in fat digestion, we now realize that they also play a much larger role in the human body. So they're involved in host microbe interactions as well as key host signaling processes. And so with this project that I'll share with you today, we really aim to expand our chemical and biological knowledge of bile acids by first synthesizing previously unknown bile acids, and then searching for them in public metabolomics data within GNPS. And then if they're present, identifying exactly where they're found. And so to do this, what we did was we created a new workflow termed synthesis-based reverse metabolomics. And in this framework, we first perform combinatorial synthesis to generate complex mixtures of potential metabolites. And then from there, we use LC-MS-MS to obtain MS-MS spectra for all of our desired products within these mixtures. And then the MS-MS spectra can be searched against all of public data in GNPS using the tool which Peter very clearly explained and introduced to you, and that tool is MAST. So what MAST does is it allows us to have an input of an MS-MS spectra, and it searches against all of public data. And then what you get out of that is a list of all the public data files where your spectra of interest are found. And so what we're able to do from these searches is get a presence-absence type of feature table on the repository scale, where we see which public data files contain each spectra of interest. And then from here, we're able to overlay sample information for data files that have redo compatible metadata, meaning that standardized ontologies were used to describe the sample information for these files. So this last step is really, really key as it enables us to start associating spectra or metabolites of interest with health-related phenotypes, as well as with organisms and sample types. So we're able to see the distribution of our compounds of interest across species or tissue types, for example. And so what we did was we wanted to apply this workflow to deblur the picture for bile acids. And specifically, we were interested in amino acid conjugated bile acids, which was a class of molecules discovered by Rob Quinn, as Peter mentioned, in our lab. And we postulated that these molecules would likely be widespread in vertebrates. And so to do this, we took 8 different bile acid cores, 4 of which are trihydroxylated and 4 of which were dihydroxylated. And then we put them each in separate reaction flasks. And then we added a mixture of 22 different amino acids to each of these reaction mixtures, both proteogenic and a few non-proteogenic as well. And this generated a total of 176 different possible bile amidate structures. So if you're keen on bile acid chemistry, you might notice that many of the bile acids used in these reactions are actually isomers of one another. So for example, all of the trihydroxylated bile acids have the same molecular formula, and the same holds true for the dihydroxylated bile acids as well. And so it's important to note at this point that one caveat to our reverse metabolomics strategy is that it's blind to stereo and regiochemistry. And this is because, typically MS/MS spectra of isomers are indistinguishable. Now, if you look close enough, you might be able to differentiate the spectra between isomers, but for this initial screen, we just wanted to concatenate isomeric compounds and simply classify each as either dihydroxylated or trihydroxylated. But I will say that including all these different isomers into our synthetic mixtures does serve a purpose. So it allows us to get authentic standards, or to have authentic standards for isomeric differentiation later on down the line if needed. And I'll highlight this use case in this talk. So overall, what we obtained was MS/MS spectra for all but 2 of those desired bile acid amidates. And we searched the spectra against GNPS public data using MAST. And we got lots of good spectral matches. So here I'm just showing you some examples of spectral matches to our alanine conjugated bile acids that we synthesized. And so what we got overall was 16,000 spectral matches to our synthesized bile acids in public data. And this was over 6,500 different unique data files that contain these compounds. So for comparison's sake, spectral matches to glycine and taurine conjugates were found in just over 27,000 unique files. So while these conjugated bile acids that we synthesize aren't quite as widespread as glycine and taurine, they are frequently observed in public data nonetheless. And then we collected the data for our synthetic mixtures on both a QTOF as well as an Orbitrap. And we compared the results of these, and what we saw was that they have a very similar number of spectral matches, and they also have similar distributions of amino acid conjugations. But really why I'm bringing this up to you as the audience is because I want to share with you that all of the data that we collected on these mixtures is publicly available through GNPS libraries. So you are able to take your own metabolomics data sets and search for these compounds within them. Okay, but what we wanted to do was gain information about where these synthesized bile acids were found in public data, and this was using redo metadata, as I mentioned just a bit ago. So what we discovered was that the majority of these compounds are present in humans, and they're also present in mice. And many of them are detected in some other vertebrates as well. And here I want to note that the color I'm using in this heat map represents the relative proportion of matches for each bile acid that are found in a particular organism, where yellow indicates the most frequent of matches and blue, or white rather, indicates the least frequent. And so with this in mind, you'll start to notice that different organisms have different patterns of bile acids. So for instance, lysine, methionine, and ornithine conjugates are found almost exclusively in mouse samples, whereas threonine conjugated bile acids are found primarily in humans. And then when we extended this type of analysis to study the distribution of these conjugated bile acids in different tissues and biofluids, what we found was that these compounds are mostly observed in gallbladder, cecum, and fecal samples. And it's interesting because only the conjugated trihydroxylated bile acids were detected in the small intestine of animals. And we also found that these synthesized bile acids are very rarely found on the skin or in liver and blood samples. But one of the most important questions that we wanted to answer with reverse metabolomics was whether these molecules were associated with particular disease phenotypes. And so what we did was we calculated the proportion of samples in each health condition that had spectral matches to our synthesized bile acids. So for instance, we found that around 80% of all IBD samples within Redo contains this glycine conjugated dihydroxylated bile acids. And then similarly, we found that many of our own synthesized conjugated bile acids were also observed quite frequently in inflammatory bowel disease patients. And it's important here to point out that this is in comparison to a low percentage of spectral matches in no disease reported. So here you can kind of compare the two. And so given these findings, what we hypothesized, given I guess that these compounds were found more frequently in IBD patients, at least with this reverse metabolomics analysis, what we did was we hypothesized that these bile acids would be detected in higher quantities in IBD patients relative to healthy controls. And so to test this hypothesis, what we did was we reached out to Dr. Ramnik Xavier at the Broad Institute, and Dr. Xavier and his colleagues have access to fecal samples from two very well phenotyped clinical cohorts of IBD patients, the IHMP2 and the PRISM cohorts. And both of these cohorts have metabolomics data that's been collected and published in the respective papers below. So we shipped our synthetic bile acid mixtures to the Broad Institute, to the Xavier lab, and they performed retention time and MS-MS matching to identify as unambiguously as possible which of our bile acids were observed in these IBD cohorts. So here I want to emphasize that this work would not have been possible without the help of all the folks whose names are underlined here. And I especially want to give thanks to Dr. Juliana Vio-Pacheco, as well as Dr. Damian Plichta, who acquired and analyzed the data respectively. Okay, so on to the data. What we found was first in the IHMP2 cohort, there were 62 matches, or there were matches to 62 of our synthesized bile acids rather. And this included 45 of which had relative quantification data. And so using this data, we found that many of our conjugated bile acids were elevated in fecal samples of people with Crohn's disease, which is shown in red here, relative to non-IBD controls shown in blue, but also relative to ulcerative colitis patients shown in yellow. And so here, those which had a statistically significant differential abundance between Crohn's or UC patients versus healthy controls are indicated by the black asterisks. And so when we performed this same type of analysis with a slightly smaller PRISM cohort, fewer of our synthesized bile acids were detected. So there were only 19 that were detected here, but the overall general trend stayed true. So in fact, we saw that most of those bile acids which were detected in the samples were elevated in Crohn's disease specifically. And so one really great thing about the method that we developed here is that it allows us to explore a particular chemical space pretty extensively and then decide which compounds within that space we want to synthesize in pure form and test for biological activity. And so here, given the marked increase of some of our conjugated bile acids in Crohn's disease patients, we synthesize those compounds that are most abundant in IBD in their pure form. And then we sent them off for testing for potential biological activities in collaboration with the Patterson lab at Penn State. And so here, this work was led by Steph Collins, which is a wonderful graduate student in the Patterson lab. And what she did was she discovered that many of these bile acids that are most abundant in Crohn's disease, specifically the glutamate conjugated bile acids, as well as threonine conjugated cholic acid, were actually potent agonists to the pregnane X receptor, or PXR. And so, in fact, several of the bile acids that she tested had activities comparable to that of the known PXR agonist rifampicin. And PXR is part of the nuclear receptor superfamily. So it's been implicated in the regulation of intestinal permeability and epithelial health. And so this is an important aspect of IBD pathophysiology, and we're interested in learning more. So Steph is now working to better understand how these compounds affect PXR signaling and host phenotype in vivo using mouse models. But after all of this work, an important question remained, which was who actually is responsible for making these compounds? And we hypothesized that they were likely produced by microbes, given that we had previously observed leucine, phenylalanine, and tyrosine conjugates in cultures of Clostridium bolte. And so what we did was, with the help of Dr. Pedro Beldefer, we set out to broadly screen human-associated bacteria for bile acid conjugation. So to do this, just over 200 bacterial isolates from the first human microbiome project were grown anaerobically in 96 well plate formats in liquid media. And then after 48 hours, media containing cholic acid and deoxycholic acid was inoculated with the live isolates, and then allowed to incubate further for 72 hours anaerobically. And then the cultures were extracted with methanol and analyzed by LC-MS. And here again, our synthetic mixtures came in handy because we were able to perform retention time and MS-MS matching to get level 1 or level 2 identification as defined by the Metabolomics Standards Initiatives of which bile acids were produced in these different isolates. So what we found was that 15 of the 22 potential amino acid conjugations could be detected in culture. And importantly, these compounds weren't detected in media or extraction blanks. And since we were able to get the phylogenetic information for all of our different strains, we were able to identify that bacteria belonging to the actinobacteria, bacilli, Clostridia, and fusobacteria classes were the main producers of these molecules, at least in culture. And so today I shared with you a story of how we've started to deblur the picture for conjugated bile acids in untargeted metabolomics. But I really look forward to seeing how this synthesis-based reverse metabolomics approach can be adapted to deblur the image of other compound classes. So specifically, our next steps are to explore the chemical space of fatty acid esters with the Siegel Lab at UCSD, in acyl amides with the Baker Lab at NC State, and some sulfated molecules with the Globitsch Lab at Uppsala University in Sweden. So I hope that you leave this talk today knowing a few critical points and a lot of these are reiterated from Peter's talk. But first, we can now efficiently search public MS-MS data using repository scale analysis tools like MAST and Redo. And this is really powerful. So I hope in the future we see a lot more use of public data. And I think we will. And I think this is a really exciting direction. So furthermore, I hope that today I guess sort of surprised you with the great diversity of bile acids. I think the diversity of bile acids is greater than previously appreciated, perhaps. And I think there's a lot more to be discovered within this class of molecules. And then finally, I hope that my story demonstrated just how powerful reuse of public data can be. And I really encourage each and every one of you to make your own data public whenever possible, so that together we can really start to deblur untargeted metabolomics data. And then last, but certainly not least, I need to give a huge thanks to my colleagues and collaborators who made this research possible. So my advisor, Peter Dorrestein, and members of the Dorrestein Lab who were instrumental in the creation and testing of this workflow. And I'd also like to thank Pedro Beldefer, who helped me out with the culturing of those microbes, and also Professor Rob Knight for allowing me to use their anaerobic chamber. And I came in as a synthetic chemist, so I really appreciate their help with training me to start culturing these microbes. And then finally, I want to acknowledge the fantastic work of my collaborators outside of UCSD. So specifically everyone at the Broad Institute who helped validate those IBD associations, as well as the Baker and Patterson labs who are helping figure out more about the biological roles of these molecules. And with that, I'd like to thank you for your attention, and we'll take any questions that you might have. Thank you.
Yeah, thank you, Emily and Peter, for really great talks and just an engaging presentation from you both about the annotation of unknowns in untargeted metabolomics data. So you have quite a few questions, but audience, please feel free to submit more questions. So I'll go ahead and start the Q&A session. Thank you both for turning on your videos. So I think this is addressed to either or, but where do tools like Sirius4 and MetFrag find their place next to annotating MSMS spectra in the GNPS environment and or using molecular networking?
Okay, now I can.
Great.
Yeah, so I wasn't able to unmute myself, so thank you for unmuting me. So MetFrag Sirius, I think they play a really good key role as well. The challenge that I often have is it's harder to verify whether those annotations. So for people who are not familiar with Sirius and MetFrag, those are in silico methods to annotate unknown information. And we widely use in silico methods in our own laboratory as well. I just didn't cover it. We just didn't cover it today. And they will play a key role. They will allow you to potentially provide a hypothesis in terms of what possible structures might be attached to those MSMS spectra. One of the challenges, of course, is how do you validate it? And that really is something that the community needs to work on quite a lot. How do we validate in silico methods? Right now, the best in silico methods, they get, and let's say you search against a structural library, they get, depending on the data set, anywhere between 10% to 50% of the annotations correct. Mm-hmm. But it's pretty hard to figure out which ones are actually which one of the matches belong to those 10%. So in silico methods are really good for hypothesis generating. Then you just need to figure out, okay, what are the follow-up experiments that we do to validate those predictions?
Yeah, and I think a follow-up question then is, should they be used complementary? I think you alluded to that, to confirm suspect formulas and or structures of unknown molecules.
Yep. Yeah, so the suspect library itself, it actually got pushed through Sirius itself. So if Sirius is able to identify a molecular formula, Sirius or Zodiac, which is another molecular formula prediction, the next generation after Sirius. So for the suspect library, we actually add the molecular formula when Sirius or Zodiac can identify the molecular formula.
Okay, clear. And this question is for Emily. Thank you for a fantastic story about bile acids. Did you test the capacity of liver enzymes of conjugated different amino acids to bile acids, except of massive work you did with microbiota species? That last part wasn't quite clear, but I think the first part of the question you got, yeah?
Yeah, I think so. So we haven't tested them with liver enzymes with the bile acid aminotransferase. We haven't we haven't performed those experiments, but we imagine that you know, they're likely not able to be conjugated with the host liver because we didn't observe these compounds. So going back to, I guess, the data set that Peter talked about, which was looking at germ-free versus specific pathogen-free mice, so colonized versus non-colonized mice, you know, you don't observe these compounds or these conjugates in germ-free mice, meaning that the host liver is likely not able to produce these, and it's the microbiota that are responsible, and the culture data kind of backs that up a little bit. So yeah.
And maybe like a follow-up to that, can you apply the finding of immunoconjugation to data already available from LCMS data, or do you have to design an experiment that enables you to do that?
Sorry, can you repeat that question?
Yeah, sure. Can you apply the finding of amino acid conjugation to data already available from LCMS data, or do you typically have to design an experiment that enables you to find it?
Okay, yeah, so this is a great question. I think for the bile acid conjugates, we're able to predict their MSMS spectra pretty well, right? Because as Peter showed, there's these very distinct losses of the amino acids. And so I think in the case of the conjugated bile acids, you know, we are likely to be able to find them in public data without using this synthesis-based reverse metabolomic strategy. But I think that for you know, in the future, right, this allows us to get authentic spectra for lots of different types of molecules, some of which might not be able to be predicted quite as easily as these amino acid conjugates. So I think, yeah, it definitely could probably be detected in public data without this synthesis, but the synthesis was really key for verifying that these were the compounds indeed, and it's also just super powerful in the future for other classes of compounds.
Thanks. And then I think a nice follow-up, what is the best platform for sharing metabolomics data?
So, you want to answer that?
I mean, I would say, you know, any platform that you can use and you can share your data on, that is great. I think any way that we can get, you know, data public, it's much appreciated. But Peter probably has
yeah. So I think for sharing your libraries, for example, I think there are a couple of really good resources where you can share your libraries and then the community can use it. Those are mass banks, the 3 mass banks. So mass bank Europe, Japan, and North America. So North America is MONA. And then the GMPS Massive that we have created. There are some other ones for plant metabolites, such as Respect, that you can share your data or your reference libraries at least. Now, what's nice is about these reference libraries is if you share it in any of those resources, they will be transferred likely to the other resources because all the resources like to use all the MSMS spectra. So just make any of these available within any of these resources, and you should be able to leverage it. Regarding your actual data deposition, so these are your metabolomics data sets. There are 3 main places where people now deposit their untargeted metabolomics data. And those are metabolites, that's largely stationed in Europe. Metabolomic Workbench, which is run out of the US, and then we have GMPS Massive, which also you can upload your data. We started to build linkages where the different repositories are slowly starting to communicate. So we created a GMPS dashboard with Metabolomic Workbench, for example, but we can also directly read out of metabolites. And we just got a BBRC NSF grant funded to improve the linkages between metabolites and GMPS massive. And so I think that will be very useful for the community. So you can deposit your data in any of these resources, and it will be used by the community. Hopefully, particularly the early members that are part of this webinar, hopefully you'll start thinking about new ways of leveraging this data. It's underutilized in my opinion, and yet there's so much information that's embedded. So.
Yeah, thanks, Peter. I think indeed it was really nice to hear how encouraging you were about the future for early career researchers to make an impact specifically in the space of utilizing various metabolomic databases.
My prediction is that we're now up to maybe 20% annotations if you include the suspect library on average. And so, and it means if you go into the ocean environment, you might get less annotations. If you go to certain types of human samples like blood or plasma, you may get more annotations. So things that are well studied. But my prediction is that within the next 10 years, this is the community that's actually going to solve the annotation problem. And that we will get easily up to 80% of all the signals annotated in the metabolomics experiment. That's my prediction. But it's this community that's going to do it.
Oh, so that's a task for the rest of us,
yes.
and those who are on the call. But I'll continue with a few more questions. So, is there a database of what the delta mass to charge value between two nodes in a molecular network that could be indicative of the molecular structure? So, for example, 90 of the substitution of glycine to phenylalanine.
Yep. So we've created a Google spreadsheet, and we're happy to share, and I'll try to find the link in a second. But the other one that I use is the Unimod, which is for protein post-translational modifications. But a lot of those protein post-translational modifications also exist in small molecules, so oxidation of a sulfur, amino acid substitution, different metals that are bound, et cetera, et cetera. So those are all a part of that list as well. And so you can look at Unimod and you'll see this list. There was recently a Twitter feed where somebody asked this particular question, and there were maybe 2 or 3 other ones that were listed where people have this particular list. Over time, this list will grow, but I will post my the link to the spreadsheet that we have in the chat in a little bit. I have to find it.
Okay, is Unimod also a tool for DIA mode? Because there's a question if you have a recommendation for the use of molecular networking tools that have for data that's acquired in DIA mode, such as MSE.
Yeah, so for GCMS, which is with EI data, it looks very from a data standpoint, it looks very similar to MSE. And basically what that means is that everything gets fragmented as it enters the instrument. What you have to do there is you first have to deconvolute all the MSMS vector, and then you can build a network of all the MSMS comparisons. Now, the challenge is most of the time, these data-independent methods don't really have a parent mass. Sometimes they do. If you have a parent mass associated with that fragment spectra, great. Then you can just use traditional molecular networking that I showed you in the talk. But the GCMS variant that uses EI type data, where you don't have a parent mass, then you may be able to use retention time instead of parent mass that you then add as the node header. So there's a Nature Biotechnology paper where we use something called MS Hub and then into molecular networking. So you can do it with MSE type data format. It's just a little bit harder to interpret because a parent mass filter is pretty informative, because the delta masses of the parent mass really helps to say, okay, this molecule is related to this molecule, let's say by 14 Dalton. You can now interpret that that's a addition of a methyl group or a CH2 group. Mm-hmm. Whereas if you don't have that parent mass information, the MS2 spectra become a little bit harder to interpret. You can still get delta MS spectra or spectra that are related, aligned. The challenge is, how do you do the interpretation? But it is possible. So this was a Nature Biotechnology paper by Alexander Askanov and with Vesakov at Imperial College that we published last year or the year before. Okay. So just look up GCMS and molecular networking in Nature Biotech, and you should be able to find it.
Great. So Emily, we have a question for you. Do the MetID methods that you used allow you to know the position on bile acid structure where amino acids conjugate?
Yeah, so I guess the short answer, I guess, is yes, somewhat. So it allows us to know the general sort of place where an amino acid, or where the amino acid conjugation takes place. So we expect, if the spectra match pretty perfectly, that it's likely the same as what's in our synthetic mixtures, right? So so we can therefore say it's likely conjugated on the carboxylic acid of the bile acid. I guess something that I mentioned during my talk, but it's important to reemphasize, is that we don't know regioisomers or stereoisomers. So for instance, if we looked at some conjugations on the hydroxyl groups of the bile acids, we couldn't say which hydroxyl group that was conjugated onto, but we are able, in this case, since there's only one carboxylic acid, and we can work through the fragmentation data to to ensure that it matches what we expected, we can say that we know where the amino acid is conjugated. But if it were another type of chemistry, that there were more positions available for that chemistry to take place, then we wouldn't be able to say exactly which position it was conjugated onto.
Thanks. A lot of nice details. So then another question for Emily. Would it be possible to include the chromatographic behavior in the reverse metabolomics approach to acid, regio, and stereoisomers? I think you can follow that one up.
Yeah. Yeah, absolutely. So so that's really how we do the confirmation work or the validation work, is using the retention times, and I think with bile acids there's pretty consistent kind of retention time shifts for those different isomers. So it is possible, like within a data set, to predict which isomers are likely seen. But I think for certainty, when we're working with any isomers, right, if you're trying to go to that level of classification, it's important to really identify with certainty which isomer it is. So this method generates those synthetic mixtures, which are really easy to pinpoint exactly which isomer is produced. So you only have 8 samples, right? It's just 8 different bile acids conjugated with 22 amino acids. So you only have to run 8 samples, right, to get a validation for 174 different bile acids. So
if you take retention time in account, you can really differentiate between a lot of the isomers. Sometimes it becomes a little bit hard. So let's say you have an leucine versus leucine conjugate. Yeah, those retention times will look very similar. And what we've done in collaboration with Aaron Baker's lab is they've run it by using ion mobility as well. So now we have both retention time as well as ion mobility information, and that can differentiate every single one of them.
Great. That was actually a follow-up question, so thanks for going ahead and answering that. And back to the instrumentation. So how much does the mass spec instrumentation and method parameters, i.e. collision energy, impact mass searches?
That's a really good question. There's a couple of things that we do to minimize that impact a little bit. So even when you have different collision energies, different instruments, the fundamental property of heating up an ion is not going to change the chemistry. It may change the intensity of some of the fragment ions that you see, but it's not going to change the chemistry of how these molecules fall apart. So that means that you have a lot of the signals that often do show up. Now, the biggest challenge, one of the bigger challenges associated with all the different instruments and even different collision energies is that you get different noise levels. And so one of the things that we do is we filter every spectra before we analyze it. And so we take 50 Dalton windows and we keep the top 6 ions in those 50 Dalton windows. And that eliminates a lot of the noise. The second thing that we do to minimize the impact of, let's say, different collision energies is we apply a square root to the spectra. What that means is that you're going to decrease the intensity of high intensity ions a little bit and increase the intensity of the low intensity ions. And so now you get a more uniform spectra, and now you do the comparisons. So when you do the comparisons, it's still within the settings that you specify. You say, I am willing to accept this much variation almost. That's one way to think about these settings. And so it may be indeed that a lot of spectra or a lot of annotations are missed, or a lot of spectral matches are missed because there's just too much variation, and thus within the criteria that you specified, they don't match. However, if you do see it, now you can look at the mirror plots and actually inspect, okay, do you believe it? That's always the first question you should ask yourself. Do you really believe it? And so Emily showed these nice mirror plots where you then have the spectra of the molecule itself and then the spectra to which it matched to. And then you have to say, okay, do I think it really is correctly annotated? And if not, then you should not include it into your analysis and maybe change your parameters so that you narrow down your search space.
And a follow-up question on the, I guess, the output for instrumentation and analysis would be, what is the relation between annotation capability and abundance? Are highly abundant metabolites identified and the unknowns are mainly low abundant ones?
So I do think on average, if you look at abundance, is that there are more of the more abundant molecules that are annotated. So that's logical because those have been structurally characterized and people have deposited those into the public domain. But Emily highlighted these bile acids, and she found them in 6,000 files, whereas the traditional host-derived metabolites, glycocholic acid and taurocholic acid, are found in 25,000 files. In many of these files, these bile acids, the new bile acids that Emily has discovered, are actually more intense than glycocholic acid and taurocholic acid, and yet they weren't originally annotated. So there's not truly a linear relationship between concentration and detectability, or annotatability, I should say. However, there is sort of a general trend that if something is more frequently observed and in large quantities, it's more likely that it's going to get an annotation, whereas things that are in low quantities, well, people might just not have seen it before or not paid attention to it before because they couldn't figure out a way to do the structural elucidation. There are literally, I imagine there's hundreds of thousands of molecules that still remain to be discovered, at least in the context of the standard traditional human metabolomics data set.
Yeah, fully agree. And do you have ideas or approaches to pursue those low abundant, unknown, unannotated species?
I mean, some of the ones that I just highlighted, or that we highlighted in this talk, propagation, using molecular networking. And here we only showed cosine-based molecular networking. But in the last year, there have been other forms of molecular networking that people have started to introduce. So there's SpecVec by the Van der Hoef lab, Simile by the Ben Bowen lab. And they build these relationships, spectral relationships, in different ways. And it may be that there are, well, actually we know that there are some patterns that are matched using those kind of scoring algorithms that are not matched based on the cosine type scoring algorithms that are used in cosine in molecular networking that is found in GMPS. Mm-hmm. And so it may be that having different algorithms that build these spectral relationships, you can begin to find different classes of molecules that you can propagate. Of course, you have the in silico methods that you can leverage. You really need to, at this point, leverage the kitchen sink. But what is most important is, once an annotation is known, yeah, I know it's kind of funny using the kitchen sink, but once you have an annotation that is known, if you can make that public, that will then help further propagate the other annotations. And this is why I think in the next 10 years, we're going to solve this problem of not being able to annotate most of the data to being able to annotate the majority of the data that is collected, because we become smarter and because we actually have data that we can build new tools with. And that's only possible if people start to share their data.
So there are two more questions left. The next is how about human exposure or xenobiotics, like environmental pollutants and pesticides? Are some of these in the suspected library? I saw you mention exposure mast.
Yeah. So basically, we've taken all public data. So if you want to look at exposure science, so these could be medications, these could be personal care products, it could be anything else from the environment. And so what is nice about these masts that we have right now for exposure, molecules related to exposure, I would use mast itself. But if you're interested in to see if anybody is exposed to something, you can mass MSMS spectra and see, are they indeed found in human data sets, and then in what sample types. So avobenzone, for example, this is a cosmetic ingredient. It's basically a sunscreen. If you want to know if that actually ends up in humans or animals environment or coral environments, all you right now have to do is mass the MSMS spectra of avobenzone and see what data sets it's found in. And then you can build a hypothesis based on the distribution of sample types, whether there is the potential for exposure happening. So, for example, you might be interested in domoic acid, which is a neurotoxin that's released by dinoflagellates, so these algal or these large dinoflagellate blooms in the ocean that kill a lot of fish. You might want to know, does that enter the food supply? All you have to do is search that MSMS, and in our case, we can actually find that it's in mackerel that is consumed by people, for example. But you can also find out where domoic acid is found throughout the world, and you will find it in many different places. So it's actually ended up being a very common molecule. So you can certainly use it for exposure science. At this point already, once we start to go into exposure masts, it will be easier to interpret the information that you get, because right now all you get is files that it match to, and then you have to go into the metadata of those data sets and figure out, okay, is this really what's the data file that is associated with that? But it will become easier for exposure science.
Good. And so lastly, Mustafa has requested a collaboration. So I am going to leave Emily and Peter's contact information here for you to reach out to them. But I'd really like to thank both Peter and Emily on behalf of the Metabolomics Society Early Member Network for giving two excellent talks today and a really great Q&A session. Please join us in the future for our webinars. But yes, thank you so much, Peter and Emily.
My pleasure, and thanks, Michelle.
Thank you
for organizing it.
Thank you.
And thank you to the audience for joining us. Yeah.
Indeed. Thanks so much.
Take care. Bye.